Good afternoon, everyone. Welcome to the CCMB and DCMB seminar today. It's a great honor to host Dr. Karen Miga's talks today. Karen Miga is the rock stars in genomics. Yeah, if you're working on genomics, you definitely heard two special issues in Nature and Science in the past 2 years in T2T genomes and the pangenome. Both are co-led by Dr. Karen Miga. Okay. Karen receives the Bachelor degree of Science in Biological Sciences, Biochemistry, and Cellular and Molecular Biologies in University of Tennessee. And later, she finished the Master degree of Science in Genetics in Case Western, not far from here. And she finished the PhD trainings in the program of Genetics and Genomics in Duke University in 2011. After that, Karen spent a couple years for the postdoc training in UCSC, University of California in Santa Cruz, and later served as the visiting scientist in University of Washington, and come back to UCSC to serve as the assistant research scientist for about 4 years until 2021. And starting from 2021, Karen served as the directors of the associate directors of the UCSC Genome Institute and assistant professors in the Department of Biomolecular Engineering department in the UCSC. And she was promoted as the associate professor and the directors of the UCSC Sequencing Technology Centers this year. However, Karen has made a lot of accomplishments and contribution to the fields of genomics and received a lot of awards. I cannot list out everything here, but I can only highlight the most important things. In 2020, Karen was recognized as one of the Nature 10 by Nature journal as the 10 people who helped shape science in the years 2020. And in the year 2022, she was named as one of the 100 most influential people of the year 2022 by the Time magazine. And she also received the Early Career Awards offered by the ASHG and also the Surveyor Scholar. So Karen has done a lot of important works in the field of genomics to set up the foundation of the next generation of the genome research, including improving the completeness of the genomic research by the T2T genomes and improving the accuracies of the genome research by the pangenome. So in both consortiums, Karen played a very important leadership roles. So let's welcome Karen for her wonderful talk today.
Thank you so much. Hopefully, can everyone hear me? Okay, I don't know if this is on. Okay, fantastic. Okay. So today I'm going to give you a bit of a whirlwind, kind of based on our introduction of thinking about where we've been as a community reaching into complete genomes, and then where we are now pushing into pangenomics. Uh-oh. No, I got it. I just did this to keep you on your toes. So when we think about genomics, we all celebrate the original human reference genome. This has really been foundational for genetic and genomic research for decades. However, we don't really think about the human reference genome as being a biochemical property necessarily. We think about it as being something that we can download on a computer no matter where you are around the world. And for decades, we've been pouring, as a global community, billions and billions of dollars into rich annotation on this information. The reason why I like to bring this up, I often teach this as my class, is I think about the human reference genome not as a string of A, T, C's, and G's, but kind of like Google Map. And we've been pouring a lot of information into this map over the years that made us very dependent on the reference genome. And in doing so, we've learned a lot of new biology. We've learned about basic science, we've learned about clinical genetics and genomics, and we're starting to learn more about human histories. However, this is a tool that we've been using, and I think we all recognize that this tool is in need of an update. Most of us have things like phones and computers. It's probably rare that anyone in this room has a phone or a computer that's 2 decades old. Yet the most important, or even biomedical instruments that we have in hospitals, right? It's very rare for us to have such a foundational resource that's so overdue for this technological update. We also know that the reference genome largely represents a single linear haplotype, And therefore, all of us in the room may not be able to make use of the same reference equally. In fact, it introduces known genetic biases that can permeate as we start to march into precision medicine. And we're also walking into an exciting new era where we know we can do better. So we should do better. And so this is really a celebration of the production of high quality genomes. We can do it. Now's the time. How can we make a better reference genome? So if we took a step back to that first slide where I kind of introduced the human reference genome as being this foundational resource. Oh, no. That's all right. I think I might have bumped it. No, I didn't that time. Can I hold it? No. Hopefully no one's got light sensitivity as strobes. I know. Here, I'll just speak when it flashes on. As you can see, yeah, exactly. Here, pay attention, then it's gonna go away. Yeah. The reference genome, of course, that we've been celebrating for so long has a ton of gaps. And we try to highlight these gaps in this bright red that's really eye-catching. And when we estimate the number of gaps that exist in the human reference genome, we're estimating about 8 to 10%, depending on whose genome you're looking at, but a minimal about 200 million bases that are missing. Do you want me to? Oh. All right. It's okay.
Oh, okay, we're gonna do our best to follow you.
That's all right. This one's where we'll start again, just because it's so easy to be captured by the red blocks. These are really the gaps that we knew existed in the human reference genome back when it was first released and celebrated as being quote unquote complete in 2003. However, we knew back then that that was really a celebration of the euchromatic portions of the human genome, not these densely heterochromatic repeat rich regions that took many decades now to resolve. And so I showed you that because it's really easy to see in our science cover what those red blocks are trying to show. These are all the gaps that had been filled by the Telomere to Telomere Consortium. This is a lovely picture of our consortium members. We were kind of online dating for a while through the global pandemic. We hadn't met in person. This is in 2022 when we all met for the first time in Santa Cruz. We were so happy to have our next face-to-face where we were able to reconvene. And in this case, it really is a celebration of these humans, not just a Karen Miga story, but a tremendous amount of individuals who came together with a mission to put together the first complete human genome. It's also a celebration of long read technologies. And so here I'm trying to really emphasize that when we first stepped into this project back in 2018, we have seen a rapid growth and development of not only PacBio in terms of establishing a firm developed product of HiFi or the high fidelity reads I'm trying to show you here, where you can sequence the same circular template many times and take a consensus and get this really highly accurate product that's typically about 20,000 bases long. We also use nanopore. When we started in 2018, it was the first paper had actually come out describing ultra long reads. So we've come quite a ways to where we now routinely use these 100,000 plus length reads that we can generate on the nanopore platform, typically on the Promethean instrument. And we also use Illumina-based data sets. And I really want to highlight this too, because people sometimes forget the importance of the short read in our work, but that's really useful for phasing. There was also a tremendous amount of energy put into the quality assessment that goes into it, all the long read tech development, but I wanted to at least use this slide to highlight two developers in terms of the assembly workflows that were kind of part of this T2T movement. One is, you might be familiar, called Virco. This was developed in Adam Phillippe's team. This has really been a workhorse for us to develop these new T2T assemblies. Another one is HiFiism, which was originally developed in and now is carried on by Haiyue Chang with ONT integration. He now has his own position at Yale. So what the goal is, of course, is to build a reference assembly where there aren't those big red blocks, but rather we think about chromosomes as being a string of text, where at the one end of the text, you're going to have a telomeric sequence, and at the other end, you're going to have a telomeric sequence, and you're going to have highly accurate base by base all the way across every single chromosome and a human genome. And what that means is that you can now introduce and start to study rich genome biology and function. We're starting to look at variation in a very comprehensive way. We're starting to now expand our catalogs of functional non-coded regions of the genome. And also by having these T2T assemblies, we've been able to kind of iterate and do rapid improvements on our methods. If we look at what we're learning, what's new. I think most of us in the audience probably understood that the acrocentric short arms of 5 of the human chromosomes were kind of completely unknown before the T2T consortium stepped in, other than, of course, the high resolution look at some of these repeat classes by fluorescence or looking in microscopy. But now we have incredibly detailed maps. And I like to include this slide because you can hopefully see the heat map. This is looking at the sequence similarity between chromosomes 21, 13, 14, 15, and 22. And you can see how dark red it is. These are extremely identical assemblies, but having them faithfully not only assembled, but error-free across, I think is quite a feat. And for each one, what we can do is we can go through and start to annotate repeat classes, as I'm trying to show you with that kind of color band that's catching your eye. We can also start looking at gene expression in these regions. We can start looking at methylation patterns in these regions. And so we're starting to build not only this map, but bring in that rich kind of on top of Google Maps, rich annotation again across these newly introduced regions. We've been able to move a little bit further, and I really want to credit Adam Philippi, who started to shift a lot of his lab in thinking about acrocentric chromosomes. What I'm showing you now is each one of those acrocentric short arms for CHM13, and I'm showing you that kind of color band structure. And what you can start to see, hopefully, is that even though they're different links, if you were to think about them as barcodes, there are shared patterns of the repeats that are all the way across. And when we started thinking about this, we're like, well, how can this new information about acrocentrics tell us something interesting or help us make predictions about things that could be happening in human, structural rearrangements and disease? And so a first place that our team has been dedicating a lot of time, so I'm hoping more papers are starting to hit by our archive soon, is expanding studies on Robertsonian translocations. So Robertsonian translocations, as you may know, are in fact involving these short arms. It has an incident rate of 1 in 1,000 births. It's the most common structural chromosomal abnormality that we see in the general population. However, if you look at all of the acrocentric Robertsonians, what you find is there's a higher incidence of seeing 13, 14, 14, 21, and 21, 21. And when we start to look at these maps, what we find is that on those particular chromosomes, 13, 14, and 21, there's a very clear shared segment, a segmentally duplicated sequence. And there's, in fact, this interesting twist where one of them is inverted relative to the others. So now we're starting to look at Robertsonian translocations to see if we can start to build more and more narratives on the genomic structure in these regions and how they could kind of facilitate some of these known chromosome abnormalities. Now, I'm in the T2T consortium because I've spent my entire life caring deeply about satellite DNA. These are the long tandem repeat classes that are found in regions of constitutive heterochromatin. And so this is a slide that I never thought I'd see in my lifetime. So it's kind of an honor to be able to share it with you. This is all of our different chromosomes from CHM13. And each of those color blocks are giving you the different satellite classes that we know exist in human centromeres. You might be able to see from the slide that every single human centromere has a red block. This is alpha satellite. This is a primate-specific 171 base pair repeat. There's no test after this, but it's the one that we typically assume is always going to be the nucleated site for the kinetochore. So that's where we expect to find a functioning centromere. Hopefully your eyes are also jumping to these massive blue and purple. These are the classical satellites that were not present in HC38. Some, for example, on chromosome 9 is over 30 megabases in length and has amazing structure. And we're just now starting to characterize some of these novel properties of some of these missing satellite classes. Now, if you're wondering where is the centromere, it doesn't occupy the entire satellite array. You could imagine, for example, the X chromosome is 3 megabases in length. Each repeat unit is 2 kilobases in length. And the kinetochore is going to sit somewhere within the context of that tandem array. And so our team was very interested to try to figure out what actually is driving the sequence component as well as what's on top. And what we found from the X work when we issued the first T2T X chromosome is that if you're marching across that array, we see we see that there are certain parts of the array that have a dip in hypomethylation. And so we call these CDRs, or centromere dip regions. Typically, when we go through and we look at chromatin IP maps as well, we find that these dips are also associated with sites that we expect to be enriched for centromeric chromatin. We now have been moving to more and more of these types of genomes. We've been building more HMMs to try to predict the sites of these CDRs. And what we're finding is the positioning of the centromere seems to be very polymorphic in the human population. So we can start to look at this now. If you have a centrally positioned, a Q arm or P arm, and start to measure the length of the array as well. We're also looking, as I'm trying to show you here, the structure of the centromere. It's not just one dip. Usually there are multi-dip domains, and that spacing and that structure could actually give us some more insights into the strength of the centromere. So our group's been trying to do more automated approaches to try to predict structure, looking at these types of centromeres as well. That's a very short vignette. I could probably do a whole talk on centromeres, but I wanted to give you kind of an overview of all these new discoveries so we could talk about pangenomics as well. One thing that was interesting when we started looking through the CHM13 genome is if you use repeat mask or any kind of repeat finder tool, you're so used to seeing lines, signs, random repeats. And when we put together the T to T genome, what we found was there were some parts of the genome where there was no repeat mask or annotation at all, like a desert. And we're like, what lives here? And sometimes what we'd find are these novel satellite classes that had never been characterized before. And sometimes there were combinations of sequences too that had repeat mask information that was going over and over again. And just kind of for the sense of giving you a feeling about them, these new Frankenstein repeats that are kind of a combination of different sequences that are then in tandem over and over and over again. And you can see hopefully by the ideogram that I'm showing that you can see all the different places with a little red it's showing again, okay, good. Where you can actually see that we're finding them not in one location of the genome, but they seem to be quite common on many different chromosomes. And these present their own kind of interesting epigenetic regulation. So this is not a centromeric satellite. This is a satellite that we don't have any clue what it's doing. And if you're marching through using epigenetic profiling, once again, what you would see is that it's highly methylated, but there are these dips that happen. And we call these methylated dip regions, you know, to really say something interesting may be happening in this particular array. We just don't understand yet. Yeah, so we're just starting to look at these methylation patterns. We've been looking at these predictions of CDRs over the centromeric satellites. And I said to my team, let's do this across the whole, every satellite array that we have in centromeric regions. We went directly to this one to see if we can repeat it. And we are finding that we can see that type of dip showing up across these 100 other haplotypes with HPRC as well. But so I'll try to talk and hopefully no one will get strobed. I know. So if you only care about satellites, like I'm a card-carrying satellite biologist, so I could just focus on those. But my colleagues in the field that love genes, I always like to point out that there are genes that are in these newly introduced regions as well. So what I'm trying to show you on the slide, if you look at chromosome 1, and if you can kind of see it projecting out, and there's like these giant blue bands, these are the parts that are still in HE38. And then there's a 20.7 million base section of new DNA, which I'm highlighting in green here. That's all new sequences. And if you look above that, you can see that kind of banding pattern where I'm kind of breaking out the blue versus red and have this arrow pointed down. And then over on the side, you can kind of see these two others. We've been teaming up with the GenCode team, trying to see, can we make new gene predictions? We're predicting new link RNAs, and we're going through with ProSeq to try to see if we can find evidence for polymerase coming through. We've been doing non-coding RNA sequencing. So these are new opportunities for us to do novel gene discovery, not only looking at things that are satellite, but actually finding points that could be hidden away in these parts of the genome as well. If we looked at a comparison of true and trusted HG38 compared to the new CHM13, all these annotations that you're seeing in red, these are all the genes that are found only in CHM13 that are not part of HG38. Any gene that's highlighted in black in this slide is present in HG38, but it's either rearranged or in a configuration that we really don't see in nature, so likely an error in HG38. This is once again crediting the work from Evan Eichler's team. Mitchell Volger was the first author, and this is giving us our first understanding of how much segmental duplication exists in the human genome. Originally, we were given numbers in textbooks of about 5%. And then after the CHM13, we've now moved this to be closer to a chromosome's worth, or chromosome size of 7% of our genome is now segmentally duplicated. And many of those segmental duplications are explaining these gene family expansions that are shown here. So in the first part of my talk, I really wanted to emphasize that these T to T assemblies, of course, have a real potential to revolutionize the way that we do genomics. When I first stepped into the space, there was a lot of excitement from our team about having these more complete maps and more comprehensive variant calls, how that could help feed into finding new variants and new discovery of understanding associations with human disease. It has more rapidly, I might say, gone into genome diversity. If you were to join our T2T community, you would not know we ever worked on human based on the number of species that we're working on now. There are T2T ruminants, T2T primate papers that are now coming out. I looked for our T2T face-to-face, and there's a T2T potato genome. So it just seems like name your favorite species. We seem to be supporting it. And it really is showing that there's this huge opportunity to study comparative genomics at the scale of whole chromosomes, which is really exciting. And where I've been finding a lot of enthusiasm, both in the T2T community and broadly, is how can we start to look at new cell biology in these newly included regions and in diploid genomes. If you think about a maternal and a paternal chromosome, it's kind of like you've duplicated that chromosome when it's in light of short reads. It's very hard to map short reads to one side or the other. And so we're starting to move into a world where a lot of genomes are diploid. And so long reads not only give us visibility or the ability to measure these new epigenetic environments in these complex regions, but they also give us an opportunity to look at allelic-based regulation of the genome. So this has been an area that we've been trying to push into. How can we start really utilizing some of the long read applications, which I won't go into today, but I'm happy to answer any questions of how that can move forward. However, back in 2022, when Adam and I were going out on the scene and we're like, the CHM13 genome is going to be great. It's going to change the way you do genetics. Everyone was like, no, thank you for your sweet enthusiasm. We will never, ever, ever leave HG38. And there's some things that I've had to learn that have kind of shaped the way that I think about even introducing the pangenome to communities that are wedded to HG38. And I will say on record, while this is being recorded, that this is being generous. Many researchers don't want to leave HG19. So I understand there are challenges with T2T genomes. One is that most of these sequences that are being newly introduced, even forgetting about the diploid component of this, just a haploid genome, are repeats. And repeats are just very difficult right now to incorporate into these standard analyses pipelines. If you're calling variants, you're calling epigenetic profiling or what have you, it's just very difficult. And even if you were to use a CHM13 assembly and to make a whole list of variant calls, you had now a VCF of all the variant calls relative to CHM13, you would have a whole list of variants that you wouldn't have any information to really build that really important annotation to let you know whether or not that did have a functional impact in your disease cohort. Much like people reference Omenn or they reference Nomad to see if it's a rare disease, here we won't have that richness to be able to provide the context to researchers to move forward. And T2T genomes are really nice, but currently they're way too expensive for folks to scale up to large cohorts. And so now you have to say, well, which T2T genome is the most useful for my study? And that makes it very hard because we see the real power of looking at variation and structural variation and all of this information at going to larger scale. I'm going to put a little vignette in the middle of my talk, just because I love satellite DNA, but I also have a very big place in my heart for sequence technology. And I think that there is a space for us to think about where technology is taking us in terms of scaling. And I thought I'd maybe just share a little bit of this now. One thing that has been kind of developing the T2T efforts is really thinking about hi-fi data and ONT data and how to use the combination of these two. However, it has, what if we had a very long read that had the hi-fi-like quality? And I think we're not quite there yet in terms of just simplex, but we're really close. So this is just showing over a very small amount of time. So this is going from 2022, and then green, you can see this is the old R9 for nanopore, where I'm showing you kind of a visual of the pore itself. And here we're looking at about Q17, at least from our lab. This is data generated just to give you some feel for in-lab versus giving something from a product page. And then if we move to R10, we can see that shift up to Q20. But now when we start using the new E821 adapter, we're starting to move into something we are calling in our lab this high accuracy simplex product, where we're starting to get closer and closer to a Q25. Also, there are error correction steps that happen through HARO. This is really crediting some amazing work coming out of Singapore, where you can do error correction on the nanopore reads, and we can see that now being shifted up to a Q40 plus. So all of a sudden now you have these reads that could be hundreds of kilobases long that are starting to push the limits in terms of really showing this amazing high accuracy like we've never seen before. And then what if we could just say, I don't need 100 KB reads in all parts of the genome. Some parts of the genome are actually easy to phase and assemble if I had a 20 KB or a 10 KB read. And so what our lab's been trying to do is think about how we can do adaptive sampling. This is where you can actually program on the nanopore which parts of the genome you want to accept and which ones you don't. Repeats are hard to work with because, you know, this mapping algorithm when you're trying to say accept or reject. But if you say, I want to reject all the easy parts of the genome and let all the hard parts come through, that's a really easy assignment. So what we're doing is we do things like that. And we're able to, all of a sudden, you can see a friendly centromere in red, right? You can see this kind of what looks like a spike, right? We're seeing this amazing enrichment over centromeres. And also we can start to get longer and longer sequence enrichment over the rDNAs here. You can see in that kind of crazy looking dot plot. This is just a single read that represents a very large adaptive sequence product from the rDNAs as well. We're also thinking about how can we do this at scale? We're not going to do 3 flow cells. It's kind of expensive for every genome. We want to make sure we can do this. So we've been working in collaboration with Benedict Payton's team to really think about how we can advance a long-read platform. So here, I'm just giving you one example of a paper that we just put out on medRxiv, where we're processing hundreds of samples and trying now to develop a protocol where we're pushing not only on ONT, 30 to 40 kilobases, but can we start to move this into what we call an ultra long, pushing more and more reads into the 100 KB plus on a single flow cell. And we're using this not only to try to do this at scale of trying to get maybe hundreds of genomes processed, but also all the downstream computational work to do the assemblies, the variant calls, and the reporting. And so that becomes this kind of seamless method. And there's a lot of interest from our team of how we can start to develop these types of method to think about personalized T2T references, not just pangenomes. We'll spend time on the pangenome part, but this is really highlighting work from Monika Chikova. Here we've been trying to think, yes, maybe one day you'll walk into a clinic, and yes, maybe one day you'll have a T2T genome. But more likely, if we're in pretend land of the future, it's going to be your whole pedigree will have their T2T genome, right? And there's a lot of power in having that type of information. And so we're trying to model that now. So if we have, for example, a 3-generational pedigree, we have the grandparents and we have the mother and we have her daughter, we can go through and build these really highly accurate assemblies. We can start to look at these personalized pedigree pangenomes, and we're starting to look at the transmission, the recombination, as well as trying to find de novo variant predictions. And it's really cool. I'm not going to go into it here out of interest of time, but we can start looking at the transmission of those CDRs we just talked about over time. We can start looking at the acrocentric short arms and how they're organized and whether or not meiotic recombination. And we're even working to see if we can see transmission of patterns of the telomeric sequences as well. So my take home from this small vignette is that I want you to leave my talk recognizing, yes, there are some really awesome things that happen in 2022. And there's a whole team of people I'm super proud to be shoulder to shoulder that announce that. But I want you to realize that you're about to be really empowered to be part of this story. Technology here is moving extremely fast. I think that you should allow yourself a moment to envision a world, maybe even within 5 years, where generating these T2T genomes is fast, it's inexpensive, and it's going to be broadly accessible. You won't need a specialist to stand in front. I used to tease the people, it won't be called a T2T genome, it's going to be called just genome. And this is something that I think if technology proves right based on what we've seen over the past few years, just be prepared. And I think it's really important for, in that context of knowing that's what we're moving into, how would this technology change the way that you do genomic research, and then just broadly, how would it change the way we do genetic research? So thank you for allowing me my little technology vignette. But that brings me back to this point, right? So when we started the T2T consortium, it was back at launch in 2018. We had the first T2TX chromosome that Adam Philippi celebrated at AGBT, and in 2019, we had the paper published on the T2TX chromosome in 2020, just to give you a place where we're starting, right? The next thing was the first complete human genome. This was the CHM13 assembly. This was in 2022. And now we're celebrating, these more and more diploid genomes with T2T in the title are coming out, but I really want to highlight what I credit to be the first T2T diploid, and that was from Adam Philippi's team, and that was HG002. They're now aiming to make this a genomic benchmark, so they're aiming for a crazy number I know this is recorded. There's such an impressive team. They're aiming for Q100, which is near perfect. And we're really building, I hope you can see by this momentum, all of a sudden now the HPRC, this Human Pangenome Reference Consortium, is on the scene in 2019. And our goal is not only to generate one genome, but to generate a whole cohort, a production of genomes, hundreds, if not thousands, of genomes that are T to T. So this is the new world. This is where the pangenome kind of unites with the first part of my talk, looking at T to T. How can we take all of this innovation? How can we take all of this technology and begin to automate it and build these impressive reference genomes to help improve that outdated reference genome of HG38 and replace it with something that represents more global diversity? And that's what's driving a lot of my lab too, to innovate in that vignette that we just went through. So I am so lucky. I am the project director and also the prime PI at the Human Pangenome Reference Consortium Genome Center at UC Santa Cruz. And so I get to be kind of in the heart of a lot of this with some amazing researchers. This was not a call to action from us. This was actually a call to action from NHGRI. It was meant to improve the representation of global genomic diversity. In that call for an RFA, there was a statement that this is kind of falling in step with the HapMap initiative and the 1,000 Genomes initiative. This is to capture common human haplotypes, things that are globally common at 1%. So this is not looking for rare disease. This is trying to get a good representation of haplotypes that are common in our population. In doing so, we organized a team of researchers with amazing expertise in long-read technologies. We've heard a lot about T to T, but I do want to put a spotlight on the VGP, the Vertebrate Genome Project, which also has leadership as part with Eric Jarvis, as well as the HGSBC with Evan Eichler as part of our key leadership as well. And so we've been working and thinking and living long-read technologies, complex variation, and T to T assemblies way before this came on the scene. We also have an amazing team of pangenomic developers. This team has really been credited for the time to shine. It's time for us to come together and start thinking about new nonlinear reference data structures and all the tooling and ecosystem that goes along with it. So this is a really big challenge. And this is going with this idea, of course, that one genome, no matter how complete and proud of it we are with these red parts filled in, is just simply not enough. And that's because we recognize that there are parts of our genome that are clinically and medically relevant that you need to have clear information on, not just one, but you need to have the variation kind of highlighted. Many of us think about examples like HLA typing, which we know can give us information about transplant matching and autoimmunity and allergies, what have you. SMA locus, once again, which is the leading cause of early infant death that we know has a lot of genomic complexity that can be captured not by one genome, but by many genomes. And also, if we think about the metabolism of drugs, they're really important sites in our genome that if we can get right, we can start to have more information. Okay. Also, these sections of the genome that we introduced with the T to T, we know these are some of the most hypervariable parts of our genome. So having one genome doesn't make a lot of sense. The centromeric regions I already showed you with the CDRs moving all around, but also the repeats in general are of different lengths. We know I'm going to time it with when it comes back. The copy number differences we know for certain gene families, like the figure I showed you with all the red genes that was really highlighting the work from Evan Eichler's team. We know this is another figure from their paper that you can start to see that there's different copy numbers of these gene families that you wouldn't capture if you were just looking at one reference genome. And also some of the really beautiful work that's been coming out of the acrocentric community, showing that now their whole chromosome arms can be interchanged that you'd miss, for example, if you didn't have these more complete maps. We also know that one genome, really it introduces biases when trying to map reads across individuals. And so this could be a problem when we're starting to be moving aggressively into concepts of genomics medicines and all kinds of initiatives. And one reason for that is not only these, interesting clinical sites of our genome, which we've been focusing on, are these newly introduced regions, but just there are some really big variants in our genome that we can't really capture that well with short reads. These, many of you are aware of, are structural variants. These are typically bigger than 50 base pairs in length. And we know that there's the estimates are tens of megabases of common polymorphic structural variants that are missing from any one individual reference. And so by not including these types of structural variants, we of course assume there's going to be biases in the human population. Now, if you did a kind of benchmark data set and you were to take short read data where you know from those particular data sets which structural variants are there, and you were to run through standard variant calling pipelines, you would find that you'd missed about 70% of these bigger structural events. So we know that there's not really a pipeline right now or a common workflow to be really comprehensive in catching bigger events. We're really good at monitoring the small ones. And so the pangenome is really taking this idea, if you take a certain individual, and here I'm showing you a T to T genome as being two lines, the gray line being maybe the chromosome you inherited from your mom, the black line being the one you inherited from your dad, and now spanning this out to be a cohort of individuals who represent diverse haplotypes. And so you can think about this as just having a series or a panel of these T to T assemblies, and then trying to define a data structure which best represents this variation. In total, we're aiming for about 1,000 haplotypes. When we think about what a pangenome is, my colleagues often remind me we can think about this in terms of 3 A's. There are a set of assemblies, there are a set of those alignments of those assemblies, and then there are the annotations on top of those rich data structure, which I'm trying to highlight here. Everything about what we're doing to enable equal use of this particular tool. Like I said at the beginning of my talk, this tool is really foundational for genetics and genomics, and we want to keep it that way. Anyone around the world with a computer can download and start working with it. Therefore, the database itself, the assemblies, the sequences are open access. We also want to create a totally open access new tooling ecosystem where anyone could have access to the methods of where we generated these assemblies, the QC, the annotation, and all of the variant calling and read alignment protocols. We also have a charge, and I think this might be one of the most difficult challenges of what we're trying to do, is people use HG38 not because its sequence is perfect, as we talked about, but because it's a one-stop shop for functional annotation. And we need to make sure we can port that into a pangenome to where that value is now that this is the hub of information that exists. And so this is, I'm just highlighting a lot of individuals here with ClinGen and Nomad, all these databases that we really value for giving meaning and annotation base by base to the reference genome. We need to see how we can start to pull that in to a pangenome database as well. So when we think about the pangenome, I always like to highlight this because this is kind of big science at its best. But I like to think that it's kind of organized in a very linear way. You can think that we have working groups where we kind of focus on population sampling and representation. We also have technology production where we bring people together who, this is their, like me, really love this stuff. You know, we really want to understand how we can be on the edge of sequencing technologies. We have organized a team to really think about T to T assembly and finishing, a whole pangenomic working group to think about the workflows and tooling. And then now I've added this extra little vignette where we're starting to move into international outreach and thinking about how to make a broader global product. Hopefully your eyes are catching on the bottom that the success of this work is really because it's embedded with a foundational embedded LC team. And so a lot of what we're doing does touch on historical perspectives, thinking about how we can move and open data sharing, how we can think about prospective recruitment, thinking about models of consent, and everything in between. We are strengthened by having these experts as part of our team, and they are embedded, which means that they contribute to each of these working groups.
New LC things did you guys find? I mean, you know, compared to the old stuff.
Oh gosh. We're developing an LC marker paper right now, so stay tuned. I think that one thing that we have as a community, we've been starting to think more and more about are topics on human iPSCs. They're thinking about the consent language of legacy lines. There's also been a lot of discussion about open access data sharing and how we can start to work with communities that maybe are more closed, have different data sovereignty models. And so that's a component. There's thinking about how you can do prospective recruitment in an urban-based manner, but yet still honor some of the tribal nations that are within the United States and how you can provide clarity and some of these recruitment into the studies. And so lots and lots and lots of things are kind of pushing in the space. But as you rightfully noted, there have been missteps in the past, like the Human Diversity Project. And I think it's one of my colleagues said we need to have HELSI, which is like adding history into it. And so we've had some really lovely scholarship in that space as well, thinking about how we can reflect on history and not repeat those missteps. Yeah. So you may ask yourself, and rightly so, who is currently represented in the pangenome? As we mentioned before, the goal of our work was to really capture common variation This is really acknowledging these past projects as well. In phase 1, we're just leaving phase 1, mind you, we only focused on the use of existing cell lines from the 1,000 genomes. And here I'm showing you all the various recruitment sites. These individuals met the mission of our moment as well, and the fact that they were properly consented for open access research. All of the resources of the biorepository, we can have access to the various cell lines. So we could go ahead and have our team step up, take the short read sequence data, hypothesize or study which of those cohorts would be most useful in terms of representing common variation, grow the cell lines up and do our checks to make sure there's no karyotype issues or anything that would present variation that we wouldn't expect to see in the human population. We also go through and try to make sure the cell lines that we're using are early passage, meaning they haven't been extensively passaged to be part of our effort as well. Our population sampling and representation work, we really have been thinking this is a kind of a harder problem. How do we begin to prioritize or rank order 1,000 genomes in order to be part of this particular project? I'm just listing a couple here just to give you a feel for it. We have a computational method where we actually have variants that have been called from these lines prior to our work, where we can have a greedy algorithm, we call it a max-var algorithm, to maximize the coverage of common variants to increase the diversity. We also have something called a max-div argument. Here you can kind of see that you have a little bit more of looking at haplotype structures. So we're trying to select based on nodes that maximize our coverage of structure as well. Now we understand that the 1,000 genomes is not sufficient to claim coverage of our planet in terms of global common haplotypes. And so we also have an active space right now where we're thinking about outreach. How do we start to do more inclusion, partnering with underrepresented populations? And this is really at this point, because we don't have that kind of sequence information based on self-reporting and geographical data. In phase 2, we are moving into prospective recruitment. So now we're no longer working with 1,000 genomes lines. We have partnered with a biobank in New York known as the BioMe Biobank. I'm really highlighting a dear friend, Emer Kinney, who's one of the PIs of our project. The Mount Sinai BioMe Biobank is pretty spectacular because it has over 70,000 participants from 160 countries, really celebrating the richness of different histories and peoples of New York. Also, we've partnered with Washington University in St. Louis as a recruitment center. They had a previous recruitment study, and we've been partnering with them through phase 1, thinking about how to model consents for open access. And this really gives us an opportunity with a lot of really talented folks who think deeply about PopGen, about how we can study shared haplotypes over time. Here, I'm just showing you a little cartoon going back, and you can start to see these things. And so this is really nice. How can we take these assemblies now and using reference panels? Can we not think about your genome as having one population descriptor, but yet you have kind of a beautiful quilt of ancestry, right, with different blocks of haplotypes. And so we're trying to think about the reference assemblies by trying to identify these types of haplotype structures across. And this is really highlighting some really lovely work by Alex, who's part of our team. And if you'd like to learn more, there's a paper here. And then we can go through and we can begin to look. This is just chromosome 22. You can start to see how you can begin to break down chromosome 22 into these geographical locations as well. The nice thing is you can label with anything you want. You can take any type of population descriptors from an area and make word clouds. It doesn't force you to use any one language, any one term, any one reference panel. It just enables tooling to begin to classify and characterize blocks that we see passing over human time. Great. So I'm going to group these together because we talked a lot about it at the beginning of my talk with technology production and thinking about phased finished T2T assemblies. I already introduced this, that we have amazing advancements in automated T2T assemblies, crediting always Adam and Hayu here. This gives me an opportunity, though, to really acknowledge some of our production team. With these big projects, sometimes they don't get a lot of spotlight, and they really deserve it. These are spectacular scientists that exist at many different production centers. They have been leaning in with our company partners for many years. And what we've been doing is really optimizing and stabilizing this really nice production of hi-fi, ONT ultra-long, and aluminum. And so this is really taking a multi-platform approach. We have been running now close to 200 of these automated T2T assemblies. And I should say near T2T assemblies. And I'm also making quotes here, but I really want to highlight, as you can see in blue, on this graph, hopefully you can see along the bottom, I'm giving you kind of all of the different chromosome names. And where you see blue, that's the number of times we're counting in this data set. that we see a T to T chromosome, meaning end to end. We have a telomere and telomere, and we have sequence in between. We align it to CHM13. We don't see anything in terms of it being outside of its expected size. And on red, what you can see is if we just kind of scaffold just a little bit, we can reach that T to T product as well. And so we're starting to move into this world where without doing a lot of manual curation, we're starting to actually capture a lot of this information. Now, hopefully your eyes are also catching on 13, 14, 15, 21, and 22. We already spent time on this in this talk. These are the acrocentric chromosomes, which we do not currently have a way to assemble across these rDNA arrays, which can be quite large. And so those we understand need a very special scaffolding method in order to bring them into T to T. There's some there, and that's because a lot of those rDNA arrays are actually quite small, so it's easy for us to traverse. So if you said, okay, great, you have a great production team, you're doing wonderful things, when can I have the data? We already had an alpha release. This was with our draft genome, which I'll talk about briefly. That was back in May of 2023. I'm sure of time. We also are gearing up now for our next beta improvement release. This will be at least 170 genomes. We are internally doing QC right now, and we're hoping to make that a big announcement to the public early in 2025. We're all mobilizing to establish a stable release of not only the T to T genome, but a stable release of a pangenome data structure in 2026. Speaking is kind of, this is to me, that 2026 deadline is very meaningful. And I think that's something that I hope if I'm invited back to this university, this would be a time when I'd say, let's start mobilizing, moving data sets, starting to partner and demonstrating that this is replacing an HD38 product. And so, and one of our final in 2028, kind of as we sunset this 5-year award, we really are thinking about a refinement. And here our goal by the RFA is really to generate at least 550 genomes. Now you saw 1,000 before, but that's because every genome has 2 phased haplotypes, and so you can multiply that by 2. And what I'm highlighting for you below in each of those boxes is really giving you an idea of all the different data types. And it does change over time. However, these are what we expect to be issuing for each of the release. And so when we start thinking about things like pangenomes and new workflow and tooling, this is where I get to take a real celebration on behalf of the leaders of our pangenome working group. I'm trying to highlight the working group leads here, really with the first release of a draft human pangenome reference. And so most of you are already familiar with what I mean by a pangenome, but it's always useful to have this type of cartoon where if you think you have 2 sequences that are aligned, here I'm just highlighting differences between those 2 sequences. So for example, you can see an insertion or deletion. You can see a single nucleotide variant or the FNV. You can also find sites of an inversion. And of course, you can do that with a multiple sequence alignment, or you could model that information in a string graph. And so here I'm showing you the sequence graph where you can now start to trace through the path of blue versus orange, and you can start to see that all of that variation is now modeled within this new data structure. So we can begin to do that not only for the very small sequence, but against all 47 genomes that we released in the draft. As you can imagine, some parts of our genome are not as exciting as others in terms of just having really nice alignments, but this is one that I wanted to bring up because it's particularly important because it's responsible for metabolism of about 25% of clinically used drugs. And so here you can see that the CYP2D6 gene can be highlighted by color. So the green, light blue, dark blue, this is just allowing your eyes to see the orientation of the gene from 5' to 3'. And what I'm doing is I'm putting the name of the gene next to each one of those different gene annotations so that you can be oriented. Now, because we have all of these reference assemblies, as I was showing you, drawing the trail of blue versus orange before, we can begin to draw paths through this graph. So here I'm showing you for each one of the assemblies, we can begin to draw the path of that assembly alignment, and we can start making predictions about how that particular assembly is structured in terms of its structural variation. And here you can see that when we take in that modest pangenome, which is not even 100 assemblies at this point, we can start to see that there are instant variation that exists within even this graph. We can start to look at the frequency, the count, and we can start to make hypotheses, for example, of how the metabolism would work based on the number of genes and the organization of the site. We also are starting to develop tools, and this is with this idea of a nod to trying to allow teams to begin to consider immediate adoption. Immediate seems a little bit more advantageous. I know it's going to take a long time to get to adoption, but really a lot of teams are focused on how can we maintain the the coordinate space of HG38. And so in doing this, our team is starting to think about workflows where we can use methods like VG giraffe, where you can take short read sequences and you can map them to a pangenome. We then take those aligned reads and we project them to HG38, and then we use Google Deep Variant to call variants to make sure we can move ahead. So far with this pangenome, which was only 47 genomes, it was just a start to show that we could, in fact, generate something and generate variant calls from it. We've already identified 120 megabases of new sequences, over 228 billion bases of haplotype resolved assemblies, more than 1,500 gene duplications, and more than 20 million of these small variants. And really, when I was pointing to before, one of the biggest challenges are really identifying these catalogs of structural variants. In this small cohort alone, we were able to report over 70,000 structural variant sites as well. Now, I hate to use this figure, I almost pulled it out because it's got a black box, and I have been so clear that we want to make everything open and everything clear. But what we're finding is that people want it to be really easy to use. They don't want to have to think about what the pangenome structure looks like. You know, if they trust that that structure meets the accuracy of our team, and we're really crazy about accuracy, then they can just go through and say, okay, I have data, I have read data, and I want to map it to your pangenome. And I'm so used to having a VCF output. So our goal is to just say, how can we facilitate this for your team, right? How can we build a tooling environment that makes this really accessible to you and impactful? And the goal here is that if you wanted to, for example, say, I'm so used to having reporting relative to the coordinate space of HG19, HG38, CHM13, all of those are paths within the pangenome. So you can get your coordinate reporting based on any coordinate space you would like. And that really enables a population of scientists to begin to think about how they want to have this reporting of variants. However, there is a real opportunity, I would say a missed opportunity for our team, if we don't go through the necessary protocols and deep thought about how we're going to start including all of this rich variation, all of this new information, all this structural variation that currently isn't part of any reference genome. And so that's something that we're working on now. So in the last few minutes of my talk, and I promise it's just a few. Exactly. That's exactly what it is. Mm-hmm.
What's the map done? Probably want to try to think about.
God, this is recorded. I agree. But I think that all that rich, the rich annotation of the CHM, not the HG38, there's been like, how do we lift over this information? How do we begin to make all this more available through the pangenome? But I do hope over time, as we do that refinement, there'll be less dependency on that.
I think I want to get you to think about getting rid of the black box. I mean, you know, you're
On record.
You know?
Yeah.
But for a bioinformatician, it's not a black box. It's just that people, you know, you're saying, look, if you don't want to look inside, you don't have to. We'll take care of it. That's what you're really saying.
Yeah, exactly.
So it's just kind of like, it's just like a toggled box. I mean, it's perfectly clear. I mean, you know, you're not hiding anything.
Nope. Everything's open.
Yeah.
Thank you for allowing me to say I hate using that slide again. So I know we're running out of time, so I wanted to be sensitive, but I wanted to at least give some space for something that I'm really excited about, and that's how we're moving into more of an international project. And so part of this is when we were starting, even in phase 1, there was a notion that, yes, this was a call to action by the US with NIH funding. However, this will be a global resource, and so it must be developed by and provide benefit to the broader global community. So last year we applied to, and we were fortunately selected to be a driver project for GA4GH. And we were able to do this with some really broad goals. How can we start to establish a federated alliance of international genomic partners? How can we work together to start thinking about standards, data sharing, reproducible cloud-based workflows? And importantly, how can we start to actually facilitate education, outreach, and capacity building? And so this is a huge amount of work, as you can imagine, but so incredibly important to make sure that this isn't just something that kind of is positioned over the US and has the broader benefit to the world. We also are entering into a new era I would say, of not only T to T assemblies, but new era of pangenomics. We are not the only show in town. Hopefully, after this talk, you go and you search the internet and you'll find that there are many pangenomic initiatives that are happening around the world. Some of them are open access like ours, and some of them are modeled to not be open access like ours. So we're now entering into a new ecosystem where there's momentum, right, to think about how we can integrate together into one global pangenome product. What would that look like? Also, how do we start communicating, comparing variants, and operating in a new world where pangenomic databases may be the new normal for folks? I mean, it might be that all different biobanks here, when they start to generate more and more assemblies, it's really natural to build a giant pangenome protected behind a firewall. How do we start to make these types of computational processes that can exist pangenome to pangenome? And so the role of the Human Pangenome Project, or the HPP, is really to help establish these new standards and frameworks to really improve genomic research as we move forward. Education and outreach, I always like to highlight this because this has been so much fun. This is really kind of highlighting some of our T to T assembly workshops, the first one that was developed really in partnership with our colleagues at Dunedin and New Zealand. This is our team flying over and really walking through some of the protocols for how to generate T to Ts. This is really celebrating some of the workshops that we've been hosting in partnership with the H3A BioNet team to really bring in this amazing team of scientists who are really starting to learn pangenomic tools. This was one that I just came from one recently, but this is really highlighting one in November as well, where a number of my team went down to help with training. And even thinking about long reads, it seems really a question of accessibility. However, I think that we are in an exciting time where we can start thinking about how to advance long read capacity building. And so with a funding that we established through Google Health, we now are setting up Promethean machines in Johannesburg, where we're able to actually work, and this is once again highlighting some beautiful work from Sergey Koren, from Adam Phillippe's team, how we can start to use one sequencing machine to generate T to T assemblies at scale. And so this is Josh Gardner, who's from my group, who flew down to Johannesburg to perform training. And here we're trying to work with their team in a collaborative way to think about 6 reference genomes coming from the Congo. And so I'll end my talk here with kind of that first slide where the goal is to really work globally to try to build an improved reference genome and position this in a way where it is once again this important data structure that can be downloaded openly and accessibly anywhere you are in the world with internet. And we're hoping that that will become the new foundational tool that will drive once again our understanding of clinical basic science as well as understanding human histories. And I like to always end this way because I feel like, of course, our project is incredibly technical. A lot of our goals are to how do we broaden studies of genetic variants, how do we think about medical treatments and risk, and how to think about personalized medicine. However, I think the last part of my talk is just as impactful because I think the legacy of our project will be defined not only by these technical achievements, but by the number of patients, clinicians, and researchers around the world who benefit from their creation and use. So with that, I'm the only person standing here, but I represent a massive team of experts. And so this along the top are all the schools that contribute and institutes to the Pangenome Project, and along the bottom is equally important are these amazing company partners who've been with us from the very beginning. And I'll be happy to take any questions if we have time. Thank you.
Thank you, Karen, for a wonderful talk. Question time.
Is there a publication or preprint for Xuanhai Lu's work on CYP2D6?
That's part of our draft pangenome paper that came out in May of 2023. I have it at the little cover here, this one.
Yeah, thank you. And can you go back to the red and blue histogram with fully assembled, fully T2D, and I didn't quite hear what.
Oh no, I know it's scaffolded. So these are, you can see that little, hopefully a little box where you see the chromosome where I included a little gap that's red at the top.
Yeah.
Oh, sorry. So that's where we know by the T to T automated method that there is a break there, but we have lots of evidence in the actual behind the scenes, the assembler, we call it grapholding, where we see evidence of it connecting, and also through high-C data with scaffolding, that actually should be put together. So for these cases that are highly conservative, where we know this is a very likely scaffolded chromosome, we include them. So that's why you see red there as well. Great question. Thank you.
Karen, it's very nice, stimulating as always. I want to ask about the graph representation. Do you imagine it to be hierarchical in a sense that some users want those loops to represent major, say, population groups? Others might want to have a different incarnation to only do that pedigree they care about. Others will kind of different zoom levels of the graph representation.
I always reflect on the 1,000 Genomes Project as a nice model. They generated all of this amazing data that was so useful and impactful, but also almost equally valuable were all the tools that came out of that initiative. And I think the pangenomic work that we're doing, a lot of people are maybe not using our assemblies, but using their own assemblies, maybe their pedigrees or maybe their data behind the firewall to build a private pangenome. Now, to your question, is there a way to actually unite those two? And so we are finding that there are teams that are already generating these near T2T assemblies of maybe population specific or cohort specific data sets. And what they're trying out now, if they take our publicly available data sets into their wherever they want to store their data, and they reconstruct the pangenome, including their references of interest, do they see value in that? And that's something that we haven't been testing robustly, but something that we're supporting now to see if it's useful.
Thank you.
Here, I have maybe a little bit of a left field question. So the T2T trios, they seem interesting and very valuable because they allow us to test the possibility of mitotic recombination, if I understand this correctly, right? Meiotic recombination, we can study this more easily, but for mitotic one, we need to really see the recombination between two alleles within one clonally expanded cell. So if those trios are from lymphoblastoid cell lines, potentially you could see evidence for that. And I think in cancer, we have lots of evidence that homologous alleles can recombine. So, but to my knowledge, this hasn't been shown in normal tissues.
We haven't really looked that deeply. I think that right now when we see this type of rearrangements happening, we're so cautious about error. We're trying to get this as perfect as possible. We're trying to make sure those aren't errors that are being introduced. So we're still in that process right now. But if there are things that appear to be really real, that can be supported, I think that would be something to follow up on after in terms of trying to identify this. I want to make, it's a little complicated because as you rightfully noted, the meiotic signatures are something we can trace quite easily, but these individuals were all recruited at the same time and they're very different ages and the cell lines were then established from those individuals going out. So there's lots of caveats in terms of, the somatic load, the selection that was placed on those cells, and then also the LCL establishment protocol and bottlenecking there that could also weigh into some of this too.
Thank you so much. Very inspiring talk. So I'm greedy. I have two questions. One is like very specific. When you talk about the epigenetic deep signal in the centromere, do you have ever like observed any specific repeat elements that would like contribute to those kind of deep signal? And yeah, I guess my second question would be a little bit broad. Basically, like as we know, like the human body will have like the mosaicism like level. Like either in the tissue level or like in the single cell level, like how do you handle this in your like in the T2T like assembly to represent? Do you have any like way to represent those mosaicisms?
So maybe I'll start backwards with the CHM13 assembly. At least from my point of view, there was a lot of concern going into that we would see a lot of somatic variation existing within these massive amounts of cells that we were sequencing because we really threw the kitchen sink at that assembly. And in particular, I was worried the satellites were going to be a complete mess. And so if you take the satellites now, the assembled hypotheses, and you take the reads and you realign them, as kind of this is matching that hypothesis, what we find is it's not super unstable. In fact, the vast majority of reads actually support that configuration. However, to your point, we do find occasionally these areas where we see variation presented, not as the majority of reads that would indicate error, but in a small fraction of the reads, which would indicate that there's some other variant in the population. And so for those, Rongri went through and created a GitHub for T2T, where you can go through and start to look at these somatic variant candidate positions and sequences. And even in our paper for the SynStat, we have little text of where we saw them. We didn't see like any sequences that were inflated, like a transposable element or anything like that. But there were sometimes like one repeat copy was advanced, more, but it wasn't like chaos. We didn't find, chaos happening. It was more like small changes that we were able to track. Now for the CDR regions, that was very interesting to our team. We wanted to know if there's any sequence features that could really describe those dips. If you look over, for example, the X chromosome, which I use as my little cartoon example, and all of those repeats underneath are 2 kb repeat units of higher order repeats. There's not like a completely different repeat that's underneath those dips, right? However, if you take each one of those 2 kb repeats and you now align them and build a phylogenetic tree, what you'll see is that there are positional variants that can distinguish groups of these repeats. They're not all the same, right? So there's actually structure of shared variants within the array. So now we're going to color these arrays. So now you can see in your mind that there's an array of 3 megabases, but there might be a stretch of orange, a stretch of yellow, a stretch of green. And now we're going to date these type of positional variants, because some of them have a lot more variants and some have a very few variants, and we can look at outgroups. And what we found as a trend is most of the time, except for 2 key examples, this CDR region was happening over the youngest repeats in the array. And so this created a model for our paper of an expanding. So example, we're seeing the younger repeats and they're pushing out like an onion, and we see the older layers on the outside. And even in the areas where we see these CDRs, that's where we find the highest when we compare between different assemblies, the places we find rearrangements or tandem duplications happening. So there's also a possibility for genomic instability in these regions for whatever reason. So now we're looking across all the HPRC assemblies to see if we can confirm that's the truth when we look over hundreds of new assemblies too.
Do we have any questions for student or the trainee postdoc? Okay, do you have a question?
I have a question that's probably pretty basic. I'm not a geneticist. So I was interested in the which cell lines you chose to represent in the initial run, and was curious why Southeast Asia wasn't represented. So as someone that doesn't have a genetics background, I mean, it's like a quarter of the world's population. So I was curious if it was a logistical reason or a theoretical reason or political reason. Yeah, exactly. Yeah, I was curious about it.
Gotcha.
We only, at the beginning, utilized cell lines that were available to us from the 1,000 Genomes Project. And the reason I understand that these particular recruitment sites were really identified was through individuals who wanted to support these research sites, and so there's a lot of researchers that were part of that project that knew the community and could support that particular activity more than it was driven by, we need to be here for, having total global oversight in terms of variant spectrum. And so that's why we know that there's an insufficient. Now for the first paper, when we were starting to study these 47 genomes, those were just, we didn't take a lot of care in the selection of year 3. We had our population genetics and population sampling representation team step up. And so we had a prediction of all the lines that we should include in production. So there's 350, and those just by chance were the ones that we happened to go through our production first. So they were not selected to represent the broadest collection. So that's more of a just production ordering more than it is anything that's representative. And we, this is fun. We actually don't pay attention to the population labels. So for us, we remove those. So we don't really focus on population descriptors, where you can think about each one of these genomes as presenting kind of a variant. You can think of it like a feature space of all the different variants. And we're highlighting now, you can see in your eyes, like all the common variants. And we're trying to select from that list the minimal set of genomes that maximally cover all those common variants. And we don't need to have population descriptors for that. So just by chance, if we don't get representation from a certain recruitment site, we're okay with that too for this project.
I want to take the chairs. We've been doing this seminar since 2004. Okay. There's only one person in the room other than me who was in the room in 2004. I'm looking at you, Tom. This is about the finest bioinformatics talk I've seen all these years here. Thank you.
Oh my gosh. Is this recorded? Thank you very much.
I'm sorry.
Thank you very much.
Thank you so much, Karen, for the wonderful talks. Although we still have a lot of questions, including myself, I have a lot of questions to ask as well, but we ran out of time. But in case you have questions, I think you can email Karen offline. And thanks, Karen's wonderful talk again. Thank you so much.